nào, cũng như là em quên record. Ờ, thì cả cái long read hi-fi và cái also này nó sẽ giải quyết những cái vùng challenge trong cái somatic variant detection như thế nào, thì mình sẽ cùng tìm hiểu qua cái sự trình bày từ cái nhà cung cấp PacBio, từ cái đại diện từ phía PacBio là anh Ray là chức vụ là Territory Account Manager của PacBio. Ngoài ra thì mình còn có cái sự hỗ trợ từ chị Tuệ là từ phía DKH là nhà cung cấp của PacBio ở Việt Nam. Thì tại vì cái kiến thức về cancer của em nó cũng hạn chế, nên là mọi người, nếu mà anh chị nào có thể đặt câu hỏi trực tiếp tới anh Ray luôn thì mình đặt câu hỏi, hoặc là mình cố gắng mình type cái câu hỏi của mình vào chatbox mà nó rõ một xíu để em đọc lên cho anh Ray, anh trả lời. Mà tốt nhất là mình hỏi trực tiếp luôn thì nó sẽ dễ hơn xíu. Dạ em cảm ơn mọi người. Hi Ray,
Yep.
can you hear me? You can start your presentation now. Can you share your screen?
Okay. Can you see my screen?
Yes.
Okay. All right. Okay, I will start. Okay. Thank you so much, everyone, for spending Sunday night with me to actually, go through a presentation about how, PacBio is changing the paradigm for cancer genomics, with our solution of both accurate long and short read sequencing. All right. So I think an interesting about PacBio is like we are the only company right now with both the long read as well as the short reads technology. So for the long read, we actually have our hi-fi sequencing, which actually delivered our long reads with the highest accuracy, to actually, provide comprehensive characterization of the genome, not only the genome, but in a multiome itself, genome, transcriptome, as well as epigenome, which I will talk more later on. And then we also have the short read sequencing platform called our ONSO system as well, which is based on the sequencing by binding technology. So it's for the SBB sequencing, it's 10 to 100 times more accurate than the conventional NGS approaches. So with that, right, we can actually cover a lot of different applications for cancer genomics, as you all can see on the screen here. And besides the different application as well, we can also support multiple different sample types, depending on, what you want to look at. So we can support, fresh frozen tissue, blood, FFP, and liquid biopsy on the short read side. And later on as well, I will share a little bit more about the insight or the advancement that we have for long read for FFP as well, which is very exciting. Okay, so the first section, I will talk more about hi-fi sequencing, which is our long read sequencing. So how do hi-fi actually works, right? It's actually, we usually extract high molecular weight DNA, and then we will start with this high quality double-stranded DNA. We will ligate the smart bell adapter at the site of this high quality DNA. And then after that, we will anneal and bind the DNA polymerase. And then after that, we will sequence it will actually form a circularized structure like this. And then it will actually be sequenced in repeated passes. So after that, we will actually generate, a lot of these different subreads. So all these different subreads, we will actually combine them together to form a consensus read we call the hi-fi reads. So what is so special about hi-fi reads is our hi-fi reads are, have very high accuracy of 99.9% accuracy. And then we can generate reads that are very long, up to 25 kb. So to compare with what we have for hi-fi sequencing compared to, let's say, Illumina and GS, right? You can see that for this particular study from the Precision FDA Truth Challenge, you can see that PacBio accuracy, we achieve Q33 accuracy compared to Illumina Q30. So we can say that we are as accurate as next generation sequencing right now, because I think in the past, 5 years ago, people will always say, long read sequencing is not accurate. The accuracy is, um, less accurate compared to NGS. But, right now for hi-fi sequencing, we can actually achieve up to Q33 threat score. And then besides the accuracy part as well, you can see that, compared to short read NGS, our PacBio raw reads, raw hi-fi reads, are 100 times longer compared to short read NGS. So with that, right, we enable a lot of different applications and a lot of things that NGS cannot capture on their sequencing results. Our hi-fi sequencing is able to actually deliver that. And then, of course, another thing about hi-fi sequencing as well, for our WGS, right, you can actually get your methylation directly from our WGS results. So you don't need to actually, perform a separate library prep, like a whole genome, like a bisulfite sequencing pre-treatment to actually, enable you to get methylation information. For our PacBio sequencing, when we are doing WGS itself, because we are sequencing on the native DNA itself, right? And then during the sequencing run, we actually take a movie of the entire sequencing run. So compared to short read sequencing, right? So short read actually take images. In every sequencing cycle, they will take an image of the fluorescent itself to tell you whether is it a A, is it a T, is it a ATGC itself lah. But for PacBio, what we do is we actually shoot an entire movie of the entire sequencing run. So by doing that, right, we can actually detect the differences between, how fast a nucleotide actually incorporate to the complementary strand itself. So by measuring the kinetics, right, so in layman term, like, if a particular A is actually methylated, it will take a longer time to be actually incorporated. Whereas for A without methylation, you can see that it takes shorter time to actually be incorporated. So by measuring this difference in time of how fast or how slow a particular A is incorporated, we can actually tell whether that particular base is methylated or not. So by having this particular technology, we can actually, in our whole genome sequencing run, you can, besides getting ACGT, you get 5MC as well as 6MA data as well. Yeah. So coming back to, just now I talked about, how hi-fi sequencing has its own advantage, right? It's highly accurate, it's long with 25 kb, and then it offers methylation data as well, 5MC, 6MA data from the whole genome sequencing data. So now we go back into, how this technology can actually contribute to, looking into cancer genomics research. So we know for cancer genomics, right? It's not only the mutations that happen doesn't only, the mutations that happen that causes cancer usually doesn't only happen on the DNA level itself, right? It's a multi-omics disease itself. So you want to look at the genomes, you want to look at the transcriptomes, and you want to look at the epigenomes as well to, give you a very comprehensive characterization of what is happening to the cancer cells itself. So I think the first part that I want to talk about is on the DNA level first. Okay, I just want to, on the next few slides, I will show you all some of the case studies of what, the different institutes or the different universities have been doing, that have been using our hi-fi sequencing to actually advance on cancer research itself. So for this particular first study that I'm showing here, right? So we know that, for breast cancer cell line, right? A particular cell line is like, things that you grow in the lab, right? So you probably already have tested it multiple times. For this particular SKBR3 cell line, it's one of the most important models for HER2 breast cancer. So it means that, for this particular cell line, it has been used in the lab uh, to be sequenced like multiple times on short read itself, right? So in this particular paper, they actually use our long read sequencing to actually hi-fi sequencing to actually sequence through this particular cell line, and they found that there are nearly 20,000 structural variation. Most of that which were missed by short read sequencing itself. So as you know, right, short reads are 100 to the reads that they generate is from 150 to 200 base pairs, correct? So a lot of times, because of the reads are so short, right? And then they need to use like assembly tool to assemble all these reads together. before they can, look at the genome itself. So because of that limitation, right, they actually miss a lot of these structural variations as well as, all this hard to sequence region, right? Repetitive regions as well. So using hi-fi sequencing, because our reads are so long, right? That's why we can span through all this hard to sequence structural variations. So that's why in this particular study, you can see that with hi-fi sequencing, they actually detected a lot of new variants that were not spotted by short read sequencing. And then this is also another study, that is used to actually look into infant with retinoblastoma. And then, they also have, used this, tested this particular patient multiple times using short read sequencing, but they were not able to actually identify what is actually causing this particular disease itself. So they use hi-fi sequencing to actually sequence this particular patient, and they found that there were two different somatic variations or two somatic structural variations that occurs in this RB1 gene itself, and then they were able to phase the different alleles. So this is also very important. If you're using short read sequencing, you will not be able to phase the different allele, but using long read, you are able to phase the two different alleles to the paternal and maternal allele itself, and then you are also able to actually identify where the mutation is, which allele the mutation is. So they found that in allele 1 they actually found a premature stop codon, and then in AL2, they found a fusion gene itself. So the RB1 has fused with this particular SIAH3 gene. So with that, right, two of the different alleles are actually not working for this particular RB1 gene that's actually causing this retinoblastoma in this infant. So it helps to solve a lot of different case with our long read sequencing. Okay, and then this particular case that is more recent, you can see that for this particular case itself, they actually so if you see this IGV itself, you can see that this is the normal haplotype, right? And then the bottom is actually the tumor cell itself. So there are a few observations that you can see from this IGV itself, right? So the first one, of course, if you can actually see the big deletion at the right side of the slides itself. So that is actually causing the the mutation that's happening in this particular PTEN gene. And then the second thing that you can see, right, is that there's only one allele here. So one of the allele has been lost during the mutation itself. So there's only one allele left for this particular cancer cell itself. And then the other thing, yeah, so I think the first thing that you see is loss of haplotype 1. The second thing is you can see there's a 12 kb deletion. And then the third thing you can see there's actually the promoter region for this P10 region is actually hypermethylated. So you can see that in one single, sequencing run, you can actually identify a lot of different things that is not possible to actually, how to say, short read sequencing will not be able to tell you all this information. Short read sequencing will probably will not be able to identify what is the cause of mutation and what is actually causing this particular cancer mutation itself. So things like, the big structural deletion, short read sequencing will not be able to do that. Loss of haplotype 1, you won't be able to phase on short read itself. So you will not be able to know about if there's any loss of heterozygosity or not. And then the epigenetic side as well, the methylation side as well, you need to probably run a second bisulfite sequencing to actually uh, identify this mutation. So this shows you how powerful long read or hi-fi sequencing is in to actually look into all these different um, cancer markers. Yeah, and then this is also another case study on BRCA itself. So on the BRCA gene, so you can see we are comparing. So they sequence the normal cell versus the tumor cell itself. So you can see that for the tumor cell, the first thing that you see is of course the huge deletion, like 29 kb deletion on the first haplotype, and then the second thing that you observe is there's actually a methylated region in the, the second haplotype as well, hypermethylated region on this promoter region. So by looking at this, right, so actually the particular patient doesn't have any BRCA gene at all. So this is what that is actually causing, probably that particular patient to be higher risk of getting, breast or ovarian cancer itself. So the same thing, this kind of mutation, you won't be able to actually, see it on short read sequencing. Yeah, and then of course, we all know about, there are multiple long read sequencing technology in the market as well. So this is just a slide, showing some of the comparison data on comparing our competitor to PacBio on the analysis of a particular HCC1395 cell line. So you can see that, the F1 scores are how we actually measure our accuracy and precision, right? And the higher F1 scores you have means the better performance your sequencing results is. So you can see that, with PacBio at 30 times coverage, tumor versus normal, compared to, ONT 75 times tumor versus 30 times normal, you can see that our accuracy is still much more higher. Even though, ONT is actually sequencing 2.5 times more sequencing coverage compared to PacBio, you can see that our accuracy is still much more higher compared to what ONT can actually offer. And in PacBio itself, also, we actually offer complete workflow for cancer whole genome sequencing. So we have the nanobind extraction kit to actually help you to extract high molecular weight DNA. And then if let's say you're doing, of course, whole genome sequencing, we have the SPK3 library prep to actually help you to do library prep. And then you can sequence it on the Revio or the Vega benchtop system that we have. And then, of course, on the analysis pipeline as well, we have our somatic WDL pipeline that can actually help you to, do all the variant calling, phasing, and I will show you all in the next slide. So our HiFi somatic WDL will actually offer you somatic variant detection from cancer samples, detecting somatic and germline SNPs, indels, allele-specific copy number, somatic structural variations, differentially methylated regions, mutational signatures, homologous recombination deficiency. So you have all this different information that's actually generated. And we actually generate in an interactive analysis report format as well, which you can actually see here. So it will just go through so after our run, after you run through the HiFi Somatic WDL, they will actually generate a report like this to show you, the table, or it will show you all these filtered like SNPs, indels, and and then you can actually filter through the different genes as well that you want to see. And then they will also show you the structural variations. So let's say, TP53, yeah, they will filter through for you. And then there's structural variations information as well that is generated from the table. Yeah, and I think all this different information that is actually listed down here is actually, you can actually retrieve it from the report itself. Yeah, so you don't need to generate your own report anymore. Like, for our PacBio pipeline, we actually help you to generate this kind of report to report all the different variants, SNP, indel, structural variations, methylated regions as well, in this particular report itself. Okay, so the next part. So that's, I went through the genome part, right? Like, you can actually detect all this big structural variations and I mean repetitive regions, difficult to sequence um, areas on the genome using our hi-fi sequencing. And then we can also, provide you phase result as well so that you can identify, where the mutation is in which different alleles. And then, of course, it comes together with the methylation information as well. So in the next part, I will go into the transcriptome. So we know, on so we know the basic dogma of biology, right? From DNA will be transcribed to RNA, RNA will be translated to proteins itself, right? So any mutations that happen on the genome will actually affect what is happening on the transcriptome. But the transcriptome is even more complex. You have all these different splicing, microRNA kind of interaction as well, which will actually affect the formation of different RNA isoforms. So that's why people are also looking into the complete transcriptome itself. So for long read sequencing itself, we can actually fully characterize the RNA dysregulation in cancer. So looking at RNA isoforms, fusion transcripts, and then the expression of the different genes itself, as well as the mutations that are expressed. So by using long read sequencing, you can actually look into all these different aspects for the RNA dysregulation in cancer. So I think one example that I want to show here is like, splicing events can actually, affect protein function. For example, this particular BCLXS gene, right? So the pre-mRNA will be spliced into different, you can see there's three different exons here. And then with the orange color exon here. So the presence of this particular orange color exon will actually form a protein called BCLXL. And then this particular BCLXL is actually anti-apoptotic, and then it will be overexpressed in cancer. Whereas for this particular one, it's the for the normal mRNA. So you can see that we probably need to look into the different isoform level to be able to actually look into to differentiate between the normal cells, cancer cells, as well as looking into cancer progressions, as well as resistance to chemotherapy. So so going back, right, we know that looking at isoform is very important. But why, have the existing sequencing technology not be able to actually provide enough isoform information? So we know like one single gene like this, right, will actually produce different types of mRNA isoforms. The combination of the different exons will provide you different types of mRNA isoforms, correct? So, but for short read sequencing, right, you can see that the reads that they generate are very short and very fragmented. So you can see all this. If you can see my mouse pointing, I hope you all can see it, but let me see if I can generate the pointer. Oops, do you all see my pointer? Okay, but yeah, I just going back to the slides just to show you like the short read sequencing will produce reads that are very short, right? So you need to actually fix it up like a Lego to tell which mRNA isoforms is it. So short-read sequencing can only assemble, 20 to 40% of the human transcriptome. It won't tell you, which isoforms you have, because you need to actually, assemble all these different fragmented reads. So in the end, you will only get a partial view of this cancer transcriptome itself. However, for long-read sequencing, on the right side, you can see that we can actually sequence through the entire mRNA. So as I mentioned, right, for hi-fi sequencing, we are very long. We are up to 25. We can generate reads that are up to 25 kb, right? mRNA is usually like 1.2 to like 2 kb itself. So we can actually sequence through the entire mRNA itself. So you don't need to actually, assemble them and then try to guess which mRNA isoform is itself. So we will just sequence through the entire thing. So that's why you can actually see a complete view of, the cancer transcriptome itself. So just, to show some examples of the studies for HiFi itself, going back to the breast cancer itself also, again, because, breast cancer is so well studied, right? So for this particular study itself, you can see that, for this particular, they actually, detected up to 142,000 of splice isoforms. And then 67% of these isoforms are actually, novel when they actually use our long read sequencing to actually sequence through the transcriptome itself. So there are 67% of the isoforms that was not detected on short read sequencing itself. So this show you how powerful hi-fi sequencing is. And then you can see that, for the genes, for example, like ERBB2, which is, associated with HER2, right? You can see the short read only, generate, and on the gene code database itself, right? You can see there are probably only around like 20 something over isoforms. But with long read sequencing, we are able to detect up to, more than 70 new novel isoform itself. So this shows that there are still so many things that short read sequencing have not revealed. So using long read sequencing, there are so many low hanging markers and things that you can actually detect both on the genome side as well as the transcriptome side itself. This is also another studies to show that the alternative promoter in this particular ARD1A isoform is also a biomarker to show that the different clinical outcome in gastric cancer. So with long read sequencing, because we are sequencing through the entire uh, mRNA itself, right? So we can actually sequence through the promoter region as well, and you can see that with this particular identification of this novel isoform, they are able to actually detect it as a biomarker for bad prognosis or worse survival. So by identifying all this biomarker as well, it can also be used for pharma to actually, try to identify some of the new drugs itself. Okay, so just now I talked about, the full length mRNA, right? So we know that for transcriptome as well, you can also do things like single cell RNA sequencing as well, right? So so in short read sequencing, let's say you're doing any 10x or parse bioscience sequencing, so you try to barcode the different transcriptomes from different cells to try to see what are the expression in all the different cells, right? However, if you're using short read sequencing, they will only be detecting the 3' end of the transcript itself. So just a few tenth of the base pairs from the 3' itself. So you will not be able to actually know about the isoform information as well. So isoform analysis is not possible. But using long read sequencing, as the same from the full length RNA side, we can actually sequence through the entire mRNA. So we are able to actually provide you the cancer isoform information in the single cell context. So this is also another study to show, a researcher using our uh, single cell RNA sequencing, and then they are actually comparing between two different technology. They are using short read as well as the long read isoform sequencing itself. So for the you can see this is a PCA plot, right, of the different cell type, and you, the distribution of the cell type is, you can see it's almost similar compared to short read it's similar, not almost similar compared to short read sequencing. So you just need to take the graph and you have to rotate it a bit, and then you can see that the short read and long read sequencing results are the same. And then on the right side of the slides, right, you can actually see that if you are just looking at gene-based expression, which short read sequencing can offer, uh, you can see that the IGF-1 expression is actually, similar in both the cancer cell and normal cell. But if let's say you are looking down into the isoform-based expression, you can see that there are difference in terms of expression in the class 1 and class 2 of these IGF-1 in normal cell compared to the tumor cell. So there are differences in expression. So you can see the class 2 isoform is more widely expressed in the cancer cell, whereas the class 1 is more in the normal cell. So this shows that, if you are just doing short read gene-based expression, you won't be able to tell this particular difference in the different isoform that is actually contributing to the cancer cells. Whereas if you are doing long read sequencing, you're able to look at the differences, the different isoform that is expressing in normal compared to the tumor samples itself. This is another example of, if we are using accurate long read RNA sequencing, right? So I mentioned earlier, if you remember, if you are using short read sequencing, they are only sequencing like I think 50 base pairs from the 3' end, right? So if you are just if you are looking at this particular fusion gene, So this IGF2, BP2, and then it fused together with this TESPA1 fusion gene, right? So if you are using short read to actually sequence it, they will just show you, there's actually a high level of expression for the TESPA1 fusion gene. So so because as I mentioned, right, so short read only sequence like the 3' end of this TESPA1. So you will only be able to detect this UTR region of this TESPA1 itself if you are using short read sequencing. But for long read, because we are sequencing through the entire gene, right? So you are able to actually tell, okay, there's actually there's a fusion incident that actually happened. So this particular IGF2 BP2 gene actually, fused with this TESPA1 gene itself. So you can see that the bottom part, the long read sequencing can actually, assign all the transcript to this particular novel fusion. And then, of course, on our long read RNA sequencing itself, also we can actually detect somatic mutation as well. So because I mentioned about, our accuracy is up to 99.9% accurate, right? So you can actually accurately call all this, somatic mutation on the RNA level as well. It's not only on DNA, but you can also do variant calling on RNA. And then there's another group that actually looks into the neoantigen receptor itself. So this particular, the neoantigen field has always, just looking at SNPs itself, right, because of short read limitation. But large structural variations and like isoforms are also likely to be more immunogenic as well. So by doing this particular study, they can actually, find new uh, using long read RNA sequencing, you can actually discover more robust neoantigen, to actually develop cancer vaccine. Yeah. So in terms of the RNA side as well, we do have a complete workflow. So like from the sample prep, and then we have this Kinnex library prep workflow that I will show you later. What is Kinnex library prep? And then after you prepare the library on Kinnex, you can actually sequence it on the Revio or the Vega instrument. And then once you have the isoform data, we have the pipelines available on SmartLink itself that can actually help you to do isoform discovery, tell you, how many novel isoform is there, how many known isoform is there. And then if you want to look at fusions, we have PB fusions tool as well that can actually help you to identify with fusion. So I mentioned about our Kinnex library prep workflow, right? So as I mentioned, for RNA itself, RNA is quite short, right? It's only like 1.2 or to 2 kb long, uh, usually, correct? So as I mentioned, if you all remember how long was hi-fi sequencing, so hi-fi sequencing is we can actually generate reads that are up to 25 kb long, correct? So if we are just using each well to actually sequence like 1 or 2 kb of the mRNA itself, we will be wasting a lot of the capacity of the hi-fi sequencing. So we are only sequencing 1 to 2 kb, right? So there's, like 23 kb capacity that is wasted itself. So what Kinect do is we actually combine these multiple cDNA together. What we concatenate all these multiple cDNA together into one single long read, and then we actually sequence it on our sequencer itself. So we combine, up to like 8 mRNA together on a full length RNA kit. And then on the single cell kit, we actually combine up to 16 different transcripts so that we can actually um, increase the throughput that you actually get from a sequencing cell itself. I mean in the sequencing smart cell itself, and then by doing that as well, we increase the throughput. The price will also go down, and then this enable us to actually do gene expression studies as well. Yeah, so this is, another tool that I mentioned earlier on, PB Fusion tool. They can actually, accurately call where the breakpoints are. So you can accurately identify where the exact breakpoints are using this particular tool. And then they can also form this circular diagram to actually show you the different, the fusion, like, where are all the fusions combining between the different genes. Let me go to the next section. Okay, so I should continue on. So I think so I covered both genome and transcriptome, right? And I think during the genome part as well, I talk a little bit about how we actually our whole genome sequencing data is able to give you methylation information as well, where you can see if a particular gene is hypermethylated or hypomethylated, which will eventually affect the expression of the gene, right? So the next part I will just talk about just show a more example for uh, epigenomes itself. So I think this is just to show you an example of again a particular tumor specific hypermethylation of this HOXD9 gene. So that's associated with EMT, right? So you can see that this particular tumor samples is hypermethylated not only in the promoter region, but it's also in the gene itself. You can also see regions that are hypermethylated itself. So these are additional information that you can actually get from just whole genome sequencing of a particular tumor sample. And then another new advancement from PacBio last year was a pre-treatment process we call the FiberSeq. So the FiberSeq enable the additional information we call the chromatin accessibility. In addition to genome, metallome, and transcriptome, right? With this particular FiberSeq technique, so what it does is like we actually have a metal six transferase enzymes that it will actually um, convert some of this uh, DNA that is actually accessible. I mean, yeah, there is not, that is not in the chromatin. So if you see all this big round thing, is these are all like, chromatins, right? Which are, they will entangle the DNA onto it, and then, those region might not be accessible, and then it will not be able to express, correct? And then by doing this particular process, we are able to actually uh, identify, what areas of the genome is actually accessible, you know, and by doing that, we can actually, detect all these 6MA, M6A markers itself. So, so you can see that, it's not only giving you 5MC information, ATGC. So with FiberSeq, it will actually give you this M6A base information also. So so you can see this particular study that was done by University of Washington. So they developed this particular fiber-seq technology, right? So they run a particular sample, use they through fiber-seq, and then through our normal library prep process, and then they also prepare a second library which is on RNA, and then going through the Kinex library prep that I mentioned, right? And then what they do is they pull the two different libraries together and then sequence on one Revio smart cell. So just one smart cell, yeah. So just on one Revio smart cell, they are able to actually, look at the genome information, the metallome, the chromatin epigenome, as well as transcriptome information itself. So in one single Revio smart cell, you can actually get a multi-omic data already. So this is an example of the IGV that you will actually get after you do this particular study, right? So you will be able, in one single Revio smart cell, you're able to look at the genetic variants. So you can see on the top here, there's genetic variants, and then you can see the CPG islands, the CPG metallome, and then the chromatin epigenome to see the chromatin accessibility, and then, of course, the transcriptome as well. And then of course, the most amazing thing is like you can see two different haplotypes. So the paternal and maternal allele. So the results are phased. So you are able to see, the four different ohms, and then from there, they are able to actually look into you, they can actually associate, what is happening on the phenotype event compared to what is happening on the molecular level. So they are able to correlate, correlate between, for example, the particular patient has a bilateral retinoblastomas, right? So they are able to actually know to say that, it's because of the X chromosome inactivation that's happening on the RB1 locus. that is enabled by the chromatin epigenome. So this is the reason why the particular patient has retinoblastomas, you know. So by in one single smart cell, you are able to actually generate all this different information to be able to, better characterize what is happening on the phenotype level. So this just shows you how powerful hi-fi sequencing is right now. Any question? I think that is on the epigenome part lah. So I think for the epigenome, besides 5MC, we can generate 6MA as well with the fiber-seq data. And then, with one single smart cell right now, you can do genomes, transcriptomes, as well as epigenomes to actually, provide you a complete characterization of that particular patient, whether is it rare disease or cancer genomics itself. So the next section is to talk about some of the advancement from PacBio as well. So we just recently launched a new protocol, which we call it Amplify. So what does Amplify means is like we are amplifying the genomic DNA for library prep and hi-fi sequencing itself. So if you remember the previous part where I talk about our whole genome sequencing workflow, right? We are actually, sequencing through the native DNA. So there's no PCR process or there's no amplification that happens during our normal library prep workflow. But for now, because we know that there are certain, challenging um, sample type, or sometimes people just don't get enough sample DNA to go through long read sequencing, right? So that's why we actually introduced this particular protocol called Amplify. So for Amplify itself, we can actually perform sequencing on Revio and Vega with as little as 1 nanogram of gDNA. So I think like what I mentioned, right? A lot of times, why people don't do long read sequencing is because they don't have enough samples. Because I think for even our Spark chemistry right now, we will require you to have at least 500 nanograms of genomic DNA before you can actually proceed for proceed for library prep and sequencing. So this has been 500 nanograms is already like a advancement. If 5 years ago you asked about this question, I think 5 years ago you probably need micrograms of gDNA, like maybe up to like 5 micrograms for one single run. But right now, because of the new chemistry as well, we managed to reduce our requirement down to 500 nanograms of gDNA for our normal workflow. And right now, with this particular Amplify protocol, we can actually support as low as 1 nanogram. So you can see in this particular slide, for PacBio protocol for Amplify, you need around 1 to 50 nanogram of gDNA. So there's a amplification step at the start where we actually use KOD extreme hot start DNA polymerase, a high fidelity DNA polymerase to actually do amplification at the start. And then we will also use the Twist UDI system as well to actually, prevent any PCR duplicates, to identify any PCR duplicates. So, and then after that, you have these two process at the start, and then the downstream workflow is the same with our normal library prep workflow. And then once you have your library, you can sequence it on the Revio or the Vega system. And then we also have, our Smart Link will be able to actually help you to demultiplex and to identify the PCR duplicates and also remove them from our hi-fi DEM. And then after that, you can use back our existing third-party tool, like what I shared earlier on, like the somatic WDL or like the normal WDL to do all the variant calling, facing, all those different tools. You can actually use the same tools for this workflow as well. So with that, right, with this particular protocol, which can actually support, less than 50 nanogram of gDNA. So now we can actually support things like sequencing small bodied organism, for example like small insects. Maybe you just want to sequence one particular insect, you can actually use this protocol, or things like metagenomics as well. Or of course, I think one of the most interesting topics for cancer genomics is FFPE tissues. So with this Amplify itself, we can also support FFPE tissues, which I will show in the next slide. So we know FFPE samples are very challenging, right? It's going through like the formalin treatment itself, it's highly degraded, and people always think FFP DNAs will usually be very short. So so we tried to sequence a few different samples with our Amplify protocol, and you can see that if you see this distribution chart, right, this is the read length of the DNA that you get from an FFP sample. So you can see average is around average. You can see the average read length for FFP tissue is probably like 2 kb, and then with also like long fragments up to 8 kb itself. So it's generally not as, complete as, 15 to 25 kb like what we do for fresh frozen tissue or blood samples. But there are still, fragments that are very long as well. I mean, long in terms of like 2 to 8 kb as well in the FFP samples itself. So with that, you can see that, these are just some of the metrics that we generated from our internal run. So you can see that the hi-fi yield that you usually get from FFP samples will be lower because the read length is lower itself. But what I want you to focus is on the read quality itself. You can see the read quality that we get from FFP samples are still, up to like Q50 Q score itself. And then, so what does it mean that, like what's the advantage, having to do long read sequencing for FFP samples, right? Of course, the first thing, like what I mentioned earlier on in my slides, is you can face the genome. You are able to face the paternal and maternal allele, and then you are able to actually identify where the mutations are, which, is the mutation in the trans or in the cis allele itself, and then whether a certain mutation is it in the same allele or is it in two different alleles. So that will also impact the expression of a certain gene, right? Correct. And then of course on long read itself we are able to like what I mentioned, we are able to sequence through all this like giant brick deletion, structural variation. So with that, our amplify with hi-fi sequencing can enable you to actually do a lot of this different give you a lot of these findings through this amplify, which enable you to actually run on FFP samples. Yeah, so that's the latest protocol that we actually enable on the cancer genomic side. Is that now it's now on hi-fi sequencing we don't only can support like fresh frozen tissue, hematological cancers with blood samples. So with Amplify itself we can also support FFP samples uh, also for cancer genomics. Okay, maybe I pause a while to see if there's any question.
Dạ mọi người có câu hỏi nào mà à, we have no questions. Okay, you can okay.
Okay, if no questions, then I will continue. All right, so the next section will, so I talk about, how hi-fi sequencing is helping you to do all this comprehensive characterization of a cancer sample, right? Looking at, not only the genomes, transcriptomes, epigenomes, you get so much more information from one single smart cell. You can do all this multi-omic study on hi-fi sequencing to actually completely characterize cancer genome itself. So looking at so many more data compared to, if you're doing like short read sequencing, you probably just get DNA and you can only look at SNPs and indels. But on hi-fi sequencing, you can look into like all this, besides SNP indels, you can be looking into structural variations, repetitive regions, and then pseudogenes, and then you get it in phase. You can actually phase those the different alleles, and then you also get methylation information as well from hi-fi sequencing. Okay, so the next section is to talk about our SBB sequencing side, right? So we know there are still certain application, for example, like liquid biopsy that may not be very optimum for hi-fi sequencing. So that's why, in PacBio itself, we have both short and long read as well. So for application that, requires higher sensitivity, we do have SBB sequencing that can actually help to actually support those type of application. So hi-fi sequencing is more like, to actually to provide you a comprehensive characterization of the particular cancer genome. But for application that requires high sensitivity, we have SBB sequencing to actually help to support those type of application. So for SBB sequencing by binding technology itself, right, we actually have our short read sequencer, which is shown here called the ONSO system. So the ONSO system, what's the value of our ONSO system is like we can generate bases that are, more than 90% above Q40. So I think the general benchmark right now, I think for most of the short read sequencing is probably like Q20 to Q30. But for our own source system itself, we promise our customers that we can actually generate 90 more than 90% basis above Q40 plus. Yeah, so I think with so what's the advantage, or like I mentioned about we can generate reads that are highly accurate, Q40 plus. So what does it mean, right, to the application itself? So with greater accuracy, it actually enables you to have greater sensitivity itself. So because of the high accuracy, right, you can actually have higher sensitivity to detect a particular, for example, a mutation in ctDNA. So and we know that, usually for the mutations that happen for ctDNA, the allele frequency is very low, correct? Probably like 1% or even lower than 1% itself. In ctDNA, it might be like 0.1%. So if your sequencer is not sensitive enough, you might miss some of this mutation itself. So that's why, with greater accuracy, we will be able to achieve greater sensitivity. And then you can see that, in terms of cost as well, because of the greater accuracy, which enable you to have greater sensitivity, right? So in order to achieve similar performance like the other short read sequencing, you can actually reduce the coverage required. So you don't need to sequence so much to actually achieve similar sensitivity. So by doing that, you're able to reduce your cost as well, which I will show you some of the case study later on as well. Yeah. So with higher sensitivity, right, like this means that you can actually, detect certain cancer type earlier, or things like, monitoring of cancer, like, minimal residual diseases. We can actually improve the performance of the test itself. And then, of course, with SBB technology, we are able to sequence through like difficult to sequence region like microsatellites as well. And with that, we can provide you greater confidence in your results. So yeah, just to show you all some of the what is the basis of sequencing by binding, right? So we know the conventional method that most of the labs are using are still based on sequencing by synthesis, correct? So what sequencing by synthesis works is like you will have a original strand, and then you will have a new strand that is actually we are building on the new strand, and then each of the sequencing cycle we will actually incorporate a new base with a fluorescent signal. So maybe G is blue, A is red, T is green, and then it will incorporate to the complementary strand, and then it will emit the particular signal. So after the camera record the signal, then it will actually cleave the fluorescent away as well as the blocker away, and then this particular cycle, happens again. But what are the disadvantage of this is like, what if, the polymerase doesn't cleave off the fluorescent? And then there's also this particular, additional, uh, section here, which might also cause like molecular scarring as well. So all this, will actually increase the noise that is happening during the sequencing cycle. So by having higher noise, you will actually decrease your accuracy. So but for sequencing by binding, for SBB, you can see that, what we do is we separate the interrogation step and the incorporation step. So for the interrogation step, you can see that we use a nucleotide with a blocker, And then we will actually, the base with a fluorescence will actually bind temporarily with the original strand. And then after that, we will take the signal. And then after that, we will wash away the nucleotide with the fluorescent. And then after that, we will activate the base itself by removing the blocker. And then after that, we will attach a new nucleotide without the fluorescent and with a blocker itself. So by doing that, we will not have the problem of, whether, the leftover fluorescence is not being cleaved away or like there's a molecular scar nucleotide. So we don't have those problem itself. So that's how we actually, increase our accuracy. Yeah, and just to show you some of examples that are being done internally as well. So you can see that for most of our runs, 100% of our bases are above Q40, and then this is an example run that actually we can actually achieve up to like 85% of our bases to be Q50. Yeah, so just to reiterate again, some of the benefits is like we can actually sequence through the difficult repetitive regions, and then there's no index hopping as well because of our different technique compared to SBS, and then there's also low duplication rate, and then you can also lower your input requirements, and finally, of course, like what I mentioned, because of our higher sensitivity, you can sequence lesser, so you can actually reduce the costs. This is just an example to show you, compared to SBB and SBS in a region with homopolymers, Lang. So you can see that, with our SBB technology, we can support, up to like, 25 similar nucleotides, and it's still going strong compared to, SBS where the performance is already degrading with the increased Lang in the homopolymer. So this will help in terms of like, application like MSI, mutation rate analysis, or like things like poly A tail sizing. Yeah, so this is, I mentioned about, why SBB has negligible index hopping and lower duplication rate, right? It's because that we use this rolling circle amplification protocol for template preparation for or cluster generation. So that's why we will not have index hopping issue or or duplication rate. Yeah, so just to mention again, to further elaborate again, because of our lesser, duplication rate itself, the number of redundant reads that you get for SBB is also lower compared to, the other technologies. So there's no wasted sequencing that happens. And of course, like what I mentioned, with SBB, right, you actually need to sequence less. So for example, for this, I will show you example case studies later, but just to show you some of the like liquid biopsy assays, right? For example, if you run on a SBS platform, their recommendation is probably asking you to run up to 25,000 X, right? However, because of our also higher accuracy, right, you can actually lower down the coverage, maybe down to fourfold itself. So by, lowering down the sequencing coverage, you are able to actually multiplex more samples on the sequencer. So by doing that, we can actually, reduce on the per sample costs. So just to, I talk about, how we can actually, sequence less, right? But you probably want to see like real data, correct? So we have this particular head-to-head comparison using Serasic ctDNA reference. So this particular Serasic has known variants, so they have 19 small variants, and then we run it through two different technology, the SBS and SBB. Yeah, so so if you see the image on the left, right? So if you see on this particular SBS results, and we actually spiked like 0.1% VAF of the mutation together with the wild type. You can see the results that you actually get from the SBS chemistry. Excuse me, sorry.
You can go to have a sip of water. We can wait.
Too much probably. Okay.
Okay, thank you.
Yeah, so if you see SBS, you can see that, the results that you get from the SBS, right? Even though, the mutation that we spike, you if you see the results here, you can see that there's 7 count of A, 10 count of C, 10 count of G, and then T is the wild type, right? If you are just looking at this particular result, you wouldn't know, what uh the spike mutation is, or like, what is the real mutation. However, if you look at SBB data, right, you can see very clearly, there's 0A, 0G, and there's 13 count of C. So, by doing that, you can actually accurately tell you, okay, the mutation is actually C. So this shows you, comparing to SBS, which is a bit noisy, for SBB, you can actually confidently say, what are the true variant is. And then this is also another comparison, so if let's say, we are sequencing, comparing between SBB and SBS at 12,000x coverage, I mean 12, yeah, 12,000x coverage, you can see that in like 0.05% VAF, you can see that the SBB sensitivity is much more accurate compared to what SBS can offer. And then in 0.1%, you can see that the result is so precise, it's only one line. And then we also tried to actually downsample the SBB to see that, what is the lowest coverage that we can go that is, equivalent to the performance for SBS. So you can see that, for example, for 0.05% VAF, at 6,000x, we are on par with 24,000x of SBS sequencing. And then the same thing on the VAF of 0.1% as well. So we actually need four less sequencing compared to the SBS, and then without the UMI as well. So that's why, I mentioned about, you actually have to sequence because of a high accuracy. You don't need to sequence as much as you sequence on SBS to actually get the same sensitivity. So by doing that, you can actually multiplex more sample in one single flow cell, and then that will actually help you to reduce the costs. Yeah, and also talking about the noise of the sequencer itself, right? So if you do look at this particular comparison between SBB and SBS, right? So you can see that for wild type for SBB, our the wild type is, you can see that the the sequencer is very precise, that, you don't have any noise in the wild type side. But however, if you look at SBS, right, you can see that even the wild type, you still are getting some of the signal itself to show you some observed allele frequency. So if you are getting results like the what you are getting on SBS, right? So the threshold that you have to put is going to be very high. You cannot be able to tell, whether a particular sample is actually a wild type or sample. If you do look at, anything that is below 0.1%, right? You wouldn't know whether is it real with the mutation, or is it really the wild type. But for SBB, you can see that it's very clear. The wild type is totally separated from the samples with spike mutation itself. So that's why you can actually, confidently tell whether whether for this with SBB, it's much more accurate. You can confidently tell which one actually has the mutation compared to the wild type. And yeah, and also, on the SBB itself, you can see that the results that you get is closer to what we actually spike in as well. So for example, this study, we spiked in 0.1% I mean mutation, right? So you can see that at SBB and SBS, our observed VAF is much more closer to the true positive or the 0.1% VAF compared to SBS. Yeah, so finally, just to show you all some of the, case studies that what other people are using our ONSO for. So as I mentioned, right, ONSO is very, it's the advantage is of course the accuracy of the sequencer itself, which enable a lot of highly sensitive application. So for example, liquid biopsy. So we work together with TGen. So you can see TGen is actually, using our ONSO system um, for liquid biopsy for multi-cancer early detection. And then, the pilot study, so you can see that on the left side, right? So that's the pilot study that they have done. And they saw that, the Q score for ONSO is, above Q50. Like most of the reads are, above Q50s, Q50 compared to, the SBS sequencer. And then we also work with McGill as well, that is using it for early detection of ovarian and endometrial cancer. And they are using a targeted panel as well. And you can see that the IGV view that the PacBio results is much more cleaner compared to what Illumina you can see on Illumina, right? So the Illumina side, you can see there's so many, noise in the IGV itself. Yeah, and then they also saw that there's the error rate is also much more lower in the SBB sequencer compared to the SBS. Yeah, so I think just to summarize the value of SBB, right? I think from this particular review paper, they actually show that a key challenge in the early detection of cancer is usually caused by a particular technology that is not sensitive enough and specific enough. So if let's say we have a technology that is like ONSO that has high sensitivity, and with high sensitivity it give you high specificity as well to confidently call how much percentage of the variant is there in a particular sample. This can actually help to disrupt the early cancer detection technology segment itself. So with that, I think that's the last of my slides. So, just to summarize again, for SBB sequencing, we have an on-source system that can actually generate reads that are 90% above Q40. And with that, with high accuracy, you can actually achieve higher sensitivity, and you can sequence less to actually save on cost to actually achieve the similar sensitivity like the SBS sequencer itself. Yeah. So thank you so much. If you all have any question on the short read sequencing side, please let me know.
Thanks so much for your sharing. Your presentation is very informative.